Dear student, in passing package, let us continue from 42nd question. What are minerals? What are ores? And what are that is ganj? So what are minerals? Minerals are the elements or compound present in the earth crust. So what is mineral? Mineral is the element or compound present in the earth crust. That is called mineral. Then what is ore? So ore means the minerals from which the metals are extracted profitably. What are minerals? Sorry, what are ores? Ores are the minerals from which metals are extracted profitably. If the metals are extracted profitably from the mineral, then such are called ores. What are ores? Ores are the minerals from which metals are extracted profitably. So these three definition you have to keep it in your mind because they may ask in the examination. What are minerals? The elements or compound present in the earth crust. Then what is a ore? Ore are the minerals from which metals are extracted profitably. Then what is a ganj? The earthly impurities such as mud, the earthly impurities present in ore. The earthly impurities present in ore is called, that is ganj. What is ganj? It is the earthly impurities. Which may be the earthly impurities? The earthly impurities may be mud, may be sand, or it may be organic matter. All these together, sand, mud, or organic matter, all these together called as earthly impurities. And these earthly impurities are present in ore. If the ore contains the earthly impurities, then they are called ganj. It is called as ganj. Now let us go for the next one. So let us study about the next one, that is write down the breakdown of glucose by various pathways. Now let us study how to write the various pathway for the breakdown of glucose. So let us write down the breakdown of glucose by various parts. So at the beginning what we have to write? So you have to write glucose, which is a 6 carbon molecule. So first you write down glucose, which is a 6 carbon molecule. This glucose in the cytoplasm, this glucose in the cytoplasm is converted into 3 carbon molecule, and those 3 carbon molecules are called pyruvate. So observe here, glucose is a 6 carbon molecule and pyruvate is a 3 carbon molecule, and this conversion of glucose into pyruvate, so glucose is converted into pyruvate where, that is in cytoplasm. What is a pyruvate? Pyruvate is a 3 carbon molecule. A 3 carbon molecule is called as pyruvate. So in the cytoplasm of a cell, glucose, which is a 6 carbon, is converted into 3 carbon. First you write down glucose. So under that one, 6 carbon converted into pyruvate where in cytoplasm as 3 carbon molecule. So then converting glucose into pyruvate release a small amount of energy. Then the pathway, that is then what happens then, after this one, then pyruvate will be converted into, so let us see how what the first glucose is converted into pyruvate, then further we should know how the pyruvate will be converted. So let us know how the pyruvate will be converted. So for that one you have to write the 3 pathways. So how many pathways you have to write? So you have to write the 3 pathways. So how the pyruvate is converted? So at the beginning what you have to write? At the beginning you have to write that 6 carbon molecule glucose is converted into 3 carbon molecules pyruvate in the cytoplasm of the cell and releases some small amount of energy. Further, what the pyruvate will be converted? The pyruvate will be converted in 3 parts. That is, 1, in absence of oxygen. Let us know what happens to pyruvate in absence of oxygen. and second one in lack of oxygen. If there is lack of oxygen, then what will be the path of the what the pyruvate will be converted? Then in presence of oxygen, absence of oxygen, presence of lack of oxygen, and presence of oxygen. So this is the pathway. First 6 carbon molecule in the cytoplasm is converted into 3 carbon molecule called pyruvate, releasing small amount of energy. Then next further what happens to pyruvate will be in the 3 parts. Pyruvate in absence of oxygen, what it will be it is converted into what? Pyruvate in lack of oxygen, what it will be converted into? Pyruvate in presence of oxygen, it is converted into what? So in absence of oxygen, pyruvate is converted into ethanol plus carbon dioxide. It is converted into ethanol, carbon dioxide, and releases small amount of energy. Pyruvate in absence of oxygen converted into ethanol, carbon dioxide, and small amount of energy. Then this is the 2 carbon molecule. So ethanol, C already we have studied, C2H5OH ethanol. Then pyruvate in lack of oxygen is converted into lactic acid plus energy. Again small amount of energy. Pyruvate in presence of oxygen, pyruvate in presence of oxygen is converted into, so this what I am writing now, this will takes place in the cell of our body, in each cell of our body. So first what happens in our body? Glucose in the cytoplasm of a cell, which is a 6 carbon, is converted into pyruvate, which is a 3 carbon. Then in the pyruvate from the cytoplasm moves into mitochondria. In mitochondria, in the cell in the cell of a mitochondria, that is the mitochondria in a cell, the pyruvate is converted into carbon dioxide plus water, and 32 ATP of energy is released in mitochondria in presence of oxygen. In mitochondria, pyruvate in presence of oxygen is converted into carbon dioxide, water, and energy. So this is the that is breakdown of glucose by various parts. So you have to keep it in your mind. Glucose 6 carbon, arrow mark, pyruvate 3 carbon. Then show 3 parts. In one part, pyruvate in absence of oxygen, ethanol, carbon dioxide, and energy. Pyruvate in lack of oxygen, lactic acid and energy. Pyruvate in presence of oxygen, carbon dioxide, water, and energy. So like this you have to show it. You have to practice to write the pathway, various pathway for the breakdown of glucose. This you have to practice, because coming 22-23 examination in April, I think this question may ask in the examination. Therefore you have to keep it in mind. Glucose is converted into pyruvate in cytoplasm and release a small amount of energy. Pyruvate in absence of oxygen, in lack of oxygen, in presence of oxygen. Pyruvate in absence of oxygen is converted into ethanol, carbon dioxide, and energy. Pyruvate in lack of oxygen converted into lactic acid and energy. Pyruvate in presence of oxygen is converted into carbon dioxide, water, and energy. So like this you have to write the various pathway for the breakdown of glucose. Now let us move for 44th question. Let us for 44th question. So in 44th question, write the function of HCl in stomach. So it is very important question. They may ask for 2 marks question, that is function of HCl. The first function is that it creates, what it creates? It creates acidic medium. It creates acidic medium for pepsin to act. What does it create? It creates a acidic medium so that pepsin can act in acidic medium, or just you can write it creates acidic medium. That is the first function. And the second function is that So it kills harmful microorganism. What is the second function? It kills harmful microorganisms. The second function of that is HCl in our stomach, it kills harmful microorganism. What are the functions of HCl in stomach? It creates acidic medium. Second one, it kills harmful microorganisms. It kills harmful microorganisms. So these are the two function of HCl. Then what is pyruvate? What is pyruvate? So the conversion of glucose, which is a 6 carbon molecule into 3 carbon molecule is called pyruvate. What is pyruvate? The conversion of glucose, which is a 6 carbon molecule, into 3 carbon molecule is called pyruvate. What is the name of the 3 carbon molecule? The name of the 3 carbon molecule is called as pyruvate. So what is a pyruvate? Pyruvate is a 3 carbon molecule, and this 3 carbon molecule we get by the breakdown of 6 carbon molecule, that is glucose. What is biomagnification? So at higher trophic level, the concentration of harmful substances increases. This is called biomagnification. What is biomagnification? At higher trophic level, the concentration, so I have written CONC, that means concentration. At higher trophic level, the concentration of harmful substances increases, and this is called as biomagnification. So this is very important. They may ask in the examination with example ಕೇಳಬಹುದು. ನೀವು example ಬೇಡ, ಇಷ್ಟನ್ನು ಕಲೀರಿ. What is biomagnification? At higher trophic level, the concentration of harmful substances increases. If there is increase in harmful substances at higher trophic level, it is called biomagnification. Bio means living, magnification means increasing. What is increasing? Harmful substances is increasing. Then coming to 47th question, write the general formula for alkane, alkene, alkyne. Nodi see, I will show first common CN. In all the three you write down CN, CN. Then H2N, for all the three you write down H2N, for all the three you write down H2N. Then for ANE, alkane, plus 2. Then here don't write anything. Then for alkyne, minus 2. What is the general formula for alkane? CnH2n plus 2. For alkene, CnH2n. For alkyne, CnH2n minus 2. So like this, we have to write the general formula. Then coming to the next question. So what is meant by homologous series? Very important for the examination. A series of homologous series is a series of same organic compounds. A series of same organic compounds but having a difference of CH2 between successive member is called homologous series. Yes, what is homologous series? A series of same organic compounds but having a difference of CH2 between successive member is called homologous series. So you can write example as methane CH4 C2H6. So you increase one carbon and 2 hydrogen. So here add one carbon, it will become 2. Add 2 hydrogen, it will become 6. For this carbon you add one more carbon C3, then it will become H8. So it is a series of, it is called homologous series because all the three are the series of the same organic compound, but between the successive member there is a difference of CH2. A series of same organic compound having a difference of CH2 between successive member is called as homologous series. CH4, C2H6, C3H8. So one carbon in each step you have to increase one carbon. In each step you have to increase one carbon. Then what is oxidation reaction? 49th question. So give example. The reaction of the reaction of adding of oxygen, reaction of adding of oxygen or removal of hydrogen. What is meant by oxidation reaction? The reaction of adding of oxygen or removal of hydrogen. Adding of oxygen or removal of hydrogen is called oxidation reaction. Example, 2Cu plus O2 give rise to 2CuO. What we have added here? Oxygen. Or you can give one more example. 2H2 plus O2 give rise to 2H2O. So what we have added? Oxygen. Or you can write another easy example, 2Mg plus O2. For magnesium we are adding oxygen to become 2MgO, that is magnesium oxide. So all the three examples you can keep it in your mind. 2Cu plus O2, 2CuO, 2Mg plus O2, 2MgO, 2H2 plus O2, 2H2O. All the three example you can keep it in your mind. So what is fertilization reaction? So the process of fusion of male gametes with female gametes. The process of fusion of male gametes with female gametes. The process of fusion of male gametes with female gametes to form zygote. The process of fusion of male gametes with female gametes to form zygote is called fertilization. Then let us go for 51st question. What is contraception? Name the methods. So what is meant by contraception? So contraception means the process of preventing. So what we have to prevent? The process of preventing. What we have to prevent? The process of preventing pregnancy. The process of preventing pregnancy is called as contraception. So what is contraception? The process of preventing pregnancy is called contraception. Then let us make a list of the methods, various methods to avoid contraception, that is to avoid pregnancy. Yes, again I will repeat. What is contraception? The process of preventing pregnancy is called contraception. Now let us list the method. The first method is called mechanical method. What is the first method? First method is called mechanical method, or it is also called as barrier method. So first method is called mechanical method or barrier method. And the second method is called as surgical method. Surgical method. Just to keep it in your mind, the definition of contraception and their methods. Contraception, the process of preventing pregnancy. Methods, one is mechanical method or barrier method, and the second method is called surgical method. Third method is called chemical method. Third method is called chemical method, and the fourth method is called, that is a natural method, or it is called as educative method. So these are the four methods used to avoid pregnancy or contraception. Yes, what is contraception? The process of preventing pregnancy is called contraception. Name the methods: mechanical method or barrier method, surgical method, chemical method, natural or educative method. So these are the method used to prevent pregnancy. Then coming to 52nd question, write the differences between inherited traits and acquired traits. Inherited traits, the transfer of the traits which get transferred, the trait which get transferred from parents to offspring, from parents to offspring. So what is inherited inherited traits? The inherited traits means the traits which get transferred from parents to offspring, that is called inherited traits. Then coming to acquired traits, acquired traits means an individual, a trait which an individual acquired during its lifetime. So that is called acquired traits. So what is acquired traits? A traits which an individual acquired during his lifetime is called as acquired traits. Then these inherited traits, it include genes. Inherited traits include genes. Then what is acquired traits? It will not include genes. Inherited traits, inherited traits include genes, but acquired traits will not include genes. So genes are not included in acquired traits, but genes are included in inherited traits. So these are the differences between inherited trait and acquired trait. Then coming to 53rd question, that is what are fossils? So fossils are the remains or preserved part, remains or preserved part of a dead organism, buried, dead organism buried in the earth crust, buried in the earth over a million of years, over a million of years is called as fossils. What are fossils? Fossils are the remains or preserved part of a dead organism buried in the earth over a million of years. Next one is name the methods to find the age of fossil. So fossil age can be found out by two methods. One is called relative depth method. One is called relative depth method. And the second one is called as, one is relative depth method and the second one is called carbon dating. Second one is called carbon dating. These are the two methods used to find the age of fossil. What is a fossil? The remains or preserved part of a dead organism buried in the earth over a million of years. Which are the two methods used to find the age of fossil? One is relative depth method, another one is carbon dating. So these are the two methods used to find the age of fossil. Then coming to 54th question, define refractive index. So refractive index is defined as the extent of bending, the extent of bending of light, the extent of bending of light when the extent of bending of light towards the normal or away from the normal, towards the normal or away from the normal is called refractive index. So what is refractive index? the extent of bending of light. The light will bend either towards the normal or away from the normal is called refractive index. So what is refractive index? The extent of bending of light towards the normal or away from the normal is called as refractive index. Now let us go for 55th question. So let us go for 55th question. Why POP should be stored in a moisture proof container? Because it is a 1 mark question. Because POP when 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 it gets moisture, POP when it get moisture, it becomes when it get moisture, it becomes a hard mass. It becomes what? It becomes a hard mass. Therefore, to avoid to becoming a hard mass, it should be kept in a moisture proof container. Why we have to keep POP in moisture proof container? Because if it when it gets moisture, it becomes a hard mass. So since it becomes a hard mass, to avoid of becoming hard mass, it is kept in a moisture proof container. So this is the answer for this question. Then coming to 56th question, why silver chloride and silver bromide is stored in dark bottle? Because when exposed to light, because when exposed to light, it it undergoes decomposition. It undergoes decomposition because if you expose them to light, it undergoes decomposition. To avoid decomposition, you can write even because to avoid decomposition. So answer you can write like this also, because when exposed to light, it undergoes decomposition. Or your answer you can write like this also, because to avoid. What to avoid? Because to avoid decomposition. To avoid the decomposition reaction, it is kept in a dark bottles. So this is the answer for 56th question. Then coming to 57th question. Next, let us go for 57th question. Write the steps involved in the process of photosynthesis. The first one is that first step is absorption of sunlight by SL I have written. That means sunlight by chlorophyll. So absorption of sunlight by chlorophyll is the first step. Then second step, second step is conversion of light energy into chemical energy. Conversion of light energy into chemical energy. After converting light energy into chemical energy, and then conversion of splitting. What to split? Splitting of splitting of water into hydrogen and oxygen. This is the second step. Conversion of light energy into chemical energy, and splitting of what to split? Water. So water is split into hydrogen and oxygen. Splitting of water into hydrogen and oxygen is the second step. And the last step is that hydrogen is added to carbon dioxide to give carbohydrates. What is the third step and last step? That is hydrogen is added to carbon dioxide to give carbohydrates. So these are the three steps involved in the process of photosynthesis. What is the first step? Absorption of sunlight by chlorophyll. Second one, conversion of light energy into chemical energy and splitting of water into hydrogen and oxygen. Third one, hydrogen is added to carbon dioxide to form carbohydrates. So these are the things that you have to keep it in your mind. Then coming to the 58th question, difference between pepsin and trypsin. The first difference is it acts in acidic medium. Pepsin acts in acidic medium. Then coming to the next one, that is trypsin. It acts in alkaline medium. One, if pepsin acts in acidic medium, then trypsin will act in alkaline medium. This is the first difference. Next, second one, pepsin it is secreted by pepsin is secreted by gastric gland. Who will secrete the pepsin? Pepsin is secreted by gastric gland. Then trypsin is secreted by pancreatic juice. Who will secrete the trypsin? Pancreatic juice will secrete trypsin. Then pepsin, it is secreted in stomach. Where it is secreted? It is secreted in stomach. Who will secrete? Gastric gland. Then it is secreted in small intestine. So these are the SI I have written. SI means small intestine. So yes, what are the differences between pepsin and trypsin? Pepsin, it acts in acidic medium. Trypsin, it acts in alkaline medium. It is secreted by gastric gland. Trypsin, it is secreted by pancreatic juice. Pepsin is secreted in stomach. Trypsin, it is secreted in small intestine. So these are the differences between pepsin and trypsin. 59th question, what happens if mucus is not secreted by gastric gland? So if mucus is not secreted in gastric gland, so it increases. So what does it increases? It increases acidity. What does it increases? It increases acidity. Then it causes ulcers. Then we feel discomfort. So these are the things that happens if mucus is not secreted by gastric gland. If mucus is not secreted by gastric gland in the stomach, it increases acidity. Then it causes ulcers. Then we feel discomfort. So these are the causes that if mucus is not secreted by the gastric gland. Then the next question is that why herbivores need a longer small intestine and carnivores need a short small intestine. So coming to herbivores, why it need a longer small intestine? Because it is very difficult. It is very difficult to digest. It is very difficult to digest cellulose. What is difficult? It is very difficult to digest cellulose. Therefore, it has longer small intestine. Then coming to carnivores. Carnivores has short small intestine because it is very, what is the opposite of difficult? Easy to digest. It is very easy to digest meat. It is very easy to digest meat. So herbivores having longer small intestine because it is very difficult to digest cellulose. Carnivores will have smaller small intestine because it is very easy to digest meat. So this is the answer for the 60th question. Coming to 70th question, that is write the function of cotyledon, radicle and plumule. Cotyledon will store food. What is the function of cotyledon? It stores food. What is the function of radicle? It grows as a future root. What is the function of plumule? It grows as a future shoot. So these are the functions of cotyledon, radicle, plumule. Cotyledon, store food. Radicle, future root. Plumule, future shoot. So these are the function of cotyledon, radicle and plumule. Now let us go for 60-second question, that is the last question of this video, that is write the function of the following plant hormone. So there are 5 plant hormones in plant. Those plant hormones are axin, cytokinins, gibberellins, abscisic acid and ethylene. Now let us make a list of the function of each hormone. So coming to the first one, the function of axin. The first function is that this is the function. So what is the functions? So first function of axin, let us see. It helps in, the first function is it helps in elongation of cell. It helps in elongation of cell. And second one, it helps in bending of, it helps in bending of stem. It helps in bending of stem towards sunlight. So these are the two function of axin. So very important. What is the function of axin? It helps in elongation of cell. Second one, it helps in bending of stem towards sunlight. So these are the two functions of axin. Now let us go for the next one, that is cytokinins. So cytokinins, the first function is it promotes cell division. What is the first function? It promotes cell division. So promoting cell division is the first function of cytokinins. Then second function, it promotes rapid cell division. First one is promote cell division. Now it promotes rapid cell division, where it promotes rapid cell division of fruits and seeds. So it promotes rapid cell division of fruits and seeds. So this is the second function of cytokinins. And the third function is it helps in opening of stomata. It helps in opening of stomata during daytime. Opening of stomata during daytime. These are the three functions of cytokinins. Cytokinins functions, it promotes cell division. Second one, it promotes rapid cell division of fruits and seeds. Third one, it helps in opening of stomata during daytime. Then coming to the third one, that is gibberellins. The first function of gibberellins is that it helps for the growth of stem and flowers. It helps in growth of stem and flowers. So stem and flowers, for their growth, who will help? That is gibberellins. Then the second function is that it promotes seed germination. What does it promote? It promotes seed germination. So seed germination is the last step that we have seen in reproduction of plant. That is the first function of gibberellins. It helps for the growth of stem and flowers. Second function, it promotes seed germination. Then let us go for the next type of hormone, that is called as abscisic acid. The first function of abscisic acid is it causes wilting of leaves. The old leaves will fall. Who will make the old leaves to fall? That is abscisic acid. So it causes wilting. Wilting means to fall. Wilting of leaves. So who causes wilting of leaves? That is what we call abscisic acid. That is the first one. Then second one is it prevents seed germination. If gibberellins promote seed germination, this abscisic acid will prevent it prevents the seed germination. That means seed will not germinate. And the third function is that it helps in closing of stomata. See here, cytokinins helps in opening of stomata, but abscisic acid helps in closing of stomata. Closing of stomata during night time. Which time? During night time. So this is the three functions of abscisic acid. abscisic acid, it causes wilting of leaves. Then it prevents seed germination. And the third one, it helps in closing of stomata during night time. So these are the functions of abscisic acid. And the last hormone of plant is, that is ethylene. So what are the functions of ethylene? The first one, it promotes fruit ripening. It promotes fruit ripening. The raw fruit will become ripen. Who will helps in ripening of fruits? That is ethylene. Ethylene will promote fruit ripening. That is first one. Then the second one is that it promotes aging of leaves. It promotes aging of leaves and flowers. It promotes aging of leaves and flowers. These are the two functions of ethylene. What is the first function? It promotes fruit ripening. Second function, it promotes aging of leaves and flowers. So these are the things that you have to study in these questions that you have to study in by seeing this video. So please encourage my videos. So I will in the future, in the coming days, I will post so many videos that will help you. So please subscribe to my YouTube channel. My YouTube channel is MSS Shore. So please subscribe and view my videos and encourage me, and I will definitely from my side, whatever the possibility is there, definitely I will help the students. So thank you very much.